Dear students, we have started lecture series on the topic Oncogenesis and Metabolism of Xenobiotics, and today we are going to deal with part 2 of this topic. Today's lecture is focused on studying some key points regarding significance of cytochrome P450 in catalyzing important reactions of phase 1 of metabolism of xenobiotics. And we would be emphasizing on two important aspects associated with cytochrome P450. The very first one is the mechanism of induction with examples of cytochrome P450. And the second one is the influence of polymorphisms or genetic polymorphisms on the activity of cytochrome P450s. Regarding cytochrome P450s, the first point that we are going to see is how can we name cytochrome P450s? What is the proper nomenclature for cytochrome P450s? So cytochrome P450s are named like this, and these are written as CYP1A1, for example. So in this type of cytochrome P450, CYP basically stands for cytochrome P450. This one, it shows that this cytochrome P450 belongs to member of family 1, and this capital A shows that this belongs to subfamily A, and the last one, it shows that this cytochrome P450 is the first individual member of that family. Cytochrome P450s are heme-based proteins. In our last lecture, we have studied in detail the role of heme of cytochrome P450 in catalyzing hydroxylation reaction in phase 1. Cytochrome P450s are found widely distributed in many species, even including bacterial species. These are found in highest or massive amounts or concentrations in liver and small intestine, and they seem to be present almost in all tissues. Now, when we talk about the presence of cytochrome P450 in liver, then you must remember that in liver, cytochrome P450s are found in the microsomes of liver. And as you know that microsomes are the fractions or fragments that contain smooth endoplasmic reticulum. Cytochrome P450 catalyzes different functions when it is present in different sites in the body. For example, if cytochrome P450 system is found in microsomes of hepatocytes or liver, then it is involved in detoxification mechanisms. And through this mechanism, it will be converting harmful compounds into harmless products. Now, if the same system, that is cytochrome P450, is found in mitochondria, for example, of placenta, ovaries, testicles, adrenal cortex, then this system would be involved in steroid or cholesterol biosynthesis. And there is one additional point for you regarding this concept, that cytochrome P450 system which is found in mitochondria, this is different in such a way that in mitochondria it uses NADPH-linked flavoprotein which is called adrenodoxin reductase and a non-heme iron sulfur protein which is called as adrenodoxin. There is wide substrate specificity associated with cytochrome P450, which shows that cytochrome P450s are capable enough to metabolize multiple and different xenobiotics at a time. So therefore, human body can handle thousands of xenobiotics at a time. Sixth point regarding cytochrome P450 system is this, that there is a specific type of lipid which is called phosphatidylcholine, which is associated with this cytochrome P450 system. And this is the major lipid which is found in the membrane of endoplasmic reticulum, where it regulates the functions of cytochrome P450. Up till now, we have studied that NADPH is involved in the donation of electrons to substrates in CYP450 system. But in some cases, it has been revealed that there is another hemoprotein which is called cytochrome B5 that may be involved in the donation of electrons in cytochrome 450 system. And this protein is found attached with the membranes of smooth endoplasmic reticulum. Now the eighth point, which is associated with the significance of cytochrome P450 in phase 1, is the most important of all, and that is induction of cytochrome P450. As you know that the meaning of induction is definitely to induce, to trigger, to initiate. So cytochrome P450s are mostly found in their inducible forms. Means that cytochrome P450s are found in different cells, but these are induced by some triggers, by some initiators. For example, if we administer phenobarbital, which is a drug, or maybe other drugs, then this drug may induce the hypertrophy of smooth endoplasmic reticulum. Hypertrophy, as you know, that is enlargement or increase in size. And here is hypertrophy of smooth endoplasmic reticulum. Now, when there is increased or enlargement in the size of smooth endoplasmic reticulum, there would definitely be 3 to 4 times increase in the amount of cytochrome P450. So it means that this cytochrome P450 has been induced by an inducer, and that inducer is phenobarbital. That is a drug. Now, what is the mechanism behind the induction of cytochrome P450? So whenever there is an inducer, for example, phenobarbital or maybe any other type of drug, then how can it induce cytochrome P450? So this inducer may actually increase the transcription of mRNA for making cytochrome P450. So if there is more mRNA for cytochrome P450, definitely there will be more cytochrome P450. Then the mRNA, it is formed in its stabilized form since it has been induced. So when it is stabilized, so it means that this is having its action for a longer period of time, and it can be converted into more amount of cytochrome P450. Similarly, there may be the stabilization of cytochrome P450 enzyme. So it means that it cannot be easily degraded. And again, cytochrome P450 is available for prolonged time to remain in its action. And there may be the induction of some other mechanisms as well. For example, induction can affect translation, which means that if there is more transcription of mRNA, there is more translation or formation of cytochrome P450 protein. So these are all the mechanisms which are induced by inducers, and these mechanisms may affect the synthesis of cytochrome P450 and hence the function of cytochrome P450. So induction of cytochrome P450 is basically a biochemical mechanism that underlies drug interaction. One drug can interact with another drug just because of the induction of different forms of cytochrome P450. Let us understand this concept with the help of examples. Here is the example 1 of induction of cytochrome P450. Supposedly, there is a patient who is taking anticoagulant drug called warfarin in order to fix blood clotting issues. Now remember that this warfarin is metabolized by CYP2C9, which means that when the action of warfarin is over, then this becomes inactivated by CYP2C9, or it is metabolized or degraded by CYP2C9. Now we take the same patient, and now the patient is going to be started on another drug which is called phenobarbital, because the same patient developed epilepsy, and for the treatment of epilepsy the same patient was given phenobarbital. He was previously taking warfarin and now he is taking phenobarbital, and we know now that warfarin is metabolized by CYP2C9. Now how can phenobarbital can interact with warfarin? This phenobarbital is basically an inducer of CYP2C9, which means that if this phenobarbital is administered to this patient, then it is going to increase the levels of this cytochrome P450, which is called CYP2C9. Now warfarin dose remains same. There is no change or alteration in the dose of warfarin. Now after some days, what would happen? Levels of CYP2C9 would get high 3 to 4 times, because CYP2C9 has been induced by phenobarbital. Now what will happen? This CYP2C9 can metabolize warfarin. So warfarin is going to be metabolized more and more quickly. So now there is more dose of warfarin needed for its effective function. Okay, so if phenobarbital has been discontinued, but the warfarin dose has been increased, as we have seen here that more dose is required of warfarin. So warfarin dose has been increased and phenobarbital has been discontinued. Now what will be the effect on CYP2C9 levels? This CYP2C9 levels would be low. Why? Because these have not been induced by phenobarbital, because phenobarbital has been discontinued. So when there is low level of CYP2C9, then it means warfarin will be less metabolized, because warfarin is now present in high dose and it cannot be metabolized by low levels of CYP2C9. So as a result of which the patient would be at high risk of bleeding, because warfarin is going to remain in its action for long, and this warfarin is not going to be metabolized by CYP2C9, because the levels of CYP2C9 have been decreased by discontinuation of phenobarbital. So this is how one drug can interact with another drug, and this is how side effects are associated with drugs just because of inductions of cytochrome P450s. Here we have second example of induction of cytochrome P450, and that is of CYP2E1. Cytochrome P450 2E1 is induced by ethanol consumption. So it means that ethanol is an inducer of CYP2E1. Now once this CYP2E1 is activated or induced, it can metabolize many solvents and more specifically the components or compounds that we see in tobacco smoke. And we know that these all compounds are carcinogens, that they can promote the development of cancer. So if a person is consuming ethanol, then CYP2E1 levels in that person would be high, because CYP2E levels would be induced by ethanol. And if the same person is having exposure to such compounds, then these compounds, as we know, are carcinogens, so these may be activated then, and as a result of which that person is always at increased risk of developing cancer. There are some isoforms of cytochrome P450, for example CYP1A1, which are involved in metabolizing compounds like polycyclic aromatic hydrocarbons and some compounds which are related to the same category. Now, polycyclic aromatic hydrocarbons are procarcinogens, that these are inactive carcinogens, and these are mostly inhaled by smoking. Now, there are enzymes which are called aromatic hydrocarbon hydroxylases that can metabolize polycyclic aromatic hydrocarbons by using CYP1A1. So in the lungs, these enzymes, by using CYP1A1, may convert inactive polycyclic aromatic hydrocarbons into their active forms or into their active carcinogens by hydroxylation reactions. And it has been seen that smokers have high concentrations of this enzyme in some of their cells and even in some of their tissues when we compare them from non-smokers. So therefore, smokers are always at high risk of developing cancer, and especially, as you know, lung cancer. Last point regarding cytochrome P450s is the effect of polymorphism on the activity of cytochrome P450s. There are some cytochrome P450s that have been noted to exist in their polymorphic forms. Polymorphic forms are basically genetically altered forms. And most of the cytochrome P450s that are present in their polymorphic forms show low catalytic activity. Means that polymorphic forms of cytochrome P450 are mostly non-functional or least functional. These are found in the body but cannot perform their actual function. So therefore, the people who are having polymorphic forms of cytochrome P450s would be showing different drug responses. For example, polymorphisms have been seen in CYP2D6, as a result of which CYP2D6 cannot perform its actual function. Normally, it is involved in the metabolism of debrisquin, which is an antihypertensive drug used to treat blood pressure, and there is another drug called spartine, which is used to control rhythms of heart. So normally, these two drugs are metabolized by CYP2D6. But if a person is having polymorphism in CYP2D6, and if the same person is going to administer these two drugs, so due to polymorphism in CYP2D6, there will be poor metabolism of these drugs in that person. There will be accumulation of these drugs in that person, and as a result of which, consequences which are not desired will be produced. Second example which is associated with polymorphism is CYP2A6. CYP2A6 is a type of cytochrome P450 which is normally involved in metabolizing nicotine to conatin, and as a result of which active nicotine is converted into its inactive form, which is called conatin. But if CYP2A6 is polymorphic, which means that this has been genetically modified or its structure has been altered, then this CYP2A6 won't be able to convert nicotine into conatin, as a result of which nicotine in its active form would remain high. Now these individuals, especially smokers, would smoke less because their blood and brain concentrations of nicotine would be elevated, and there is no more need of nicotine or tobacco. With this, we have finished phase 1 of metabolism of xenobiotics, and we have also covered in detail the role or significance of cytochrome P450 system in phase 1. In our next lecture, we'll be starting phase 2 conjugation reactions in detail. Okay students, activity for you. You are supposed to recall today's lecture and give the answers of these two queries. I'm going to reveal the answers. If you have answered both queries appropriately, it means you have understood the all concepts of today's lecture.